Congratulations! If you've reached this far, that means you're well on your way to completing your research capsules. This lecture will talk about biases in research. In order for us to appreciate what biases mean, we will need to discuss two important concepts, that of validity and how it differs from reliability. After which, we will be talking about systematic versus random error. Then we will discuss important biases in research and the methodologic issues to reduce your biases. This is an important concept because it is the goal of every researcher to have valid results. And by removing the possible sources of bias or controlling for this, you will be increasing your likelihood of having valid results. Let's talk about the concepts of validity and reliability. If we take this target and define the truth as the bullseye and each mark as an observation, so these are attempts to determine the truth. How can we describe these observations? These observations are close to the truth. And by definition, these are valid observations. Validity is defined as closeness to the truth. And if your observations or conclusions are close to the truth, then it becomes acceptable. In the context of quantitative results, it also refers to accuracy. Now let's take a look at another set of observations. These observations did not necessarily hit the bullseye, but you will notice that they are close to each other. And this is the concept of reliability. Reliability is also synonymous to precision or repeatability or even reproducibility. So after hearing these two concepts, you will appreciate that you have observations which are reliable but not accurate, or accurate because the mean of these observations still approximate the bullseye, but not reliable because the observations are considered far from each other. And this is what we want in our research, both accurate and reliable. And what we don't want is inaccurate and unreliable. So after having heard these two concepts, how do we now define bias? If validity is close to the truth or closeness to the truth, bias can be considered the opposite of validity. Therefore, bias can be defined as a deviation away from the truth. However, let me caution you about this definition because it lacks a qualifier. For example, if this is the truth and you have observations which are far from the truth, you cannot call this bias because these are random deviations away from the truth. In fact, they can be considered what we refer to as random error. Random error is likely to result in observations above the true value as well as below it. And if we take the mean of this random error, it will still approximate the truth. So the mean of many unbiased observations on samples still tends to correspond to the true value in the population. And this is known as noise. Bias in research is a process at any stage of inference tending to produce results that depart systematically from the true values. The key word is systematic. So bias is also known as systematic error, which is defined as a systematic deviation from the truth. So if the true value is in the center of the bullseye and your research convinces you that where the observations are is the truth, then this is what we refer to as a systematic deviation away from the truth. So you might say that bias is reliably inaccurate or reliable inaccuracy. Bias and noise are not mutually exclusive. In other words, you can have both bias and noise at the same time. So you can have results that look like this. So these are multimodal, and it shows you that there is both bias and noise. Now, in clinical observations, for example, when we look at mean arterial blood pressure measurements, using two different methods of obtaining the mean, such as using a sphygmomanometer, which is an external and indirect method of measuring blood pressure, compared to the intra-arterial transducer. This is an implant inserted into the artery to directly measure the blood pressure. You can see that there is bias. It's called a measurement bias because of using two different instruments. The difference in the means between these two methods of blood pressure measurement represents the bias, and the spread of the normal curve represents the noise. In research, there are 5 important biases that need to be accounted for. These are selection bias, performance bias, detection bias, exclusion bias, and confounding. Because in any research, whether it's an experiment or not, what you observe is the observed answer. But the observed answer is actually the true answer plus systematic error plus random error. Therefore, there is a need to control for systematic error, as we cannot really control for random error. And biases can occur in any stage of implementation. So this is a table that outlines the stage of the study, the source of the bias, the type of the bias, and how to address this. Before the implementation of your study procedure, you can have biases in the selection of participants. So if the selection of participants favors one group compared to another, this is also known as selection bias. Or if the selection of participants overestimates or underestimates the results, this is a form of selection bias called channeling bias. There's really no need to memorize channeling bias. Just consider this a form of selection bias. So how do you address this? By randomizing patients or animals and concealing the randomization. For surveys, it may be important to do a sampling procedure, which will ensure representativeness of your sample compared to the sampling frame. During the conduct of the study, there can be biases in the study procedures, including preparation of reagents and assays. So if the study procedure is different between the two groups, or ascertainment of the exposure for observational studies is different among the two groups, this can be considered performance bias. So if you decide in your experiment to do the control group this week and the experimental group next week, the reagents that you choose this week may be different in the preparation from the reagents that you prepared the next week. Also, the temperature, the humidity may affect your study animals. And that will represent a performance bias. It also tells us that when we perform our experiments, the same procedures should be strictly followed regardless of the treatment groups. In clinical studies, if there is bias in ascertainment of the exposure, for example, in a case control study, that can be considered interviewer bias. And if people cannot recall their exposures, or the likelihood that those who have the outcome of interest will recall their exposure compared to those without the outcome of interest, that is called recall bias. This can be addressed by standardizing all your study procedures or performing pilot studies so that you will perform these procedures very consistently. You can also consider having fewer people do the procedures, or in ascertainment of the exposure in a case control study, you should consider blinding or masking the assessors. If the stage of the study, the bias occurs during the outcome assessment, this is also known as measurement bias. Or if the bias occurs in the manner of ascertainment of the outcome, which may be different among groups, this is also referred to as detection bias. How is this addressed? This is addressed by standardizing all outcome assessment procedures or performing pilot studies to get used to the outcome procedures. If the outcome requires, for example, histologic section of the liver of study animals, then it would be great if only one person does this, or if you can standardize the procedure so that you are consistent in your manner of histologic staining. For outcomes that are subjective, you can consider an adjudication board to do the outcome assessment because it will always be better if several people agree on that outcome. You can also measure inter-observer viability. You need to calibrate all instruments that are going to be used for outcome assessment, and you can also consider blinding or masking the outcome assessors. If the bias occurs during the analysis of results, and this can occur if you censor or remove participants, or not including those who dropped out from the analysis of results, or not accounting for interaction for an important variable, then these types of bias can be defined as exclusion bias for censoring, attrition bias for not including those who dropped out, and confounding bias for not accounting for an important confounding factor. This can be addressed by accounting for all participants in the study and for performance of the analysis adjusted for the confounding factor. So we can say that there are four general biases in research: selection bias, performance bias, measurement bias, which is also different from detection bias, and exclusion bias, which is different from attrition bias. Biases can occur in animal studies as well. This is a study that was published in 1995 by McCanns. that show the statistical quality of published animal studies. It showed that 61% of these studies required statistical revision, 5% had serious errors, 30% had deficiencies in design, such as no randomization, size, heterogeneity, and bias. 45% had deficiencies in statistical analysis, and 33% had deficiencies in the presentation of results. In a study of papers published in PLOS ONE, published in 2009, out of 271 papers, 4% did not mention the number of animals used, 5% did not clearly state the purpose of the study, 6% did not indicate how many separate experiments were done, 13% did not identify the experimental unit, 24% reported neither the age nor the weight of the animals, 26% failed to state the sex of the animals, and 35% had discrepancies in the sample size and the numbers specified in the materials and methods. 100% had no justification of sample size. This tells us that there are certain details that should be included in your reports. These details include the objectives of the research and/or the hypothesis to be tested, the rationale for the choice of the animal or the animal model, and you need to describe the animals used in terms of species, strain, sex, age, weight, source and type of the animal, and a brief detail of the environment, diet, and husbandry. Each experiment should actually be numbered and should have the following details: the purpose of the experiment, the experimental design, the experimental unit, the method of randomization, the method of sample size determination and number of animals used, the statistical methods used for analysis, and the conclusions. Please remember this when you finish your studies. A well-designed experiment should have absence of bias, and this can be avoided by describing the experimental unit, randomization, and blinding, especially of the outcome assessors. It should also have high power, which means low noise, large sample size as much as possible, a wide range of applicability, which means that you should have replicates over other factors, simplicity, and amenable to statistical analysis. In summary, the truth can be estimated more accurately by removing bias. Bias, unfortunately, cannot be measured, but maneuvers can be included to avoid the likelihood of bias. The truth can be estimated more precisely by minimizing noise, and statistical methods can be used to minimize noise as well as a large sample size. So let me end by this quote by Thomas Hardy on bias. It says, "A well-proportioned mind is one which shows no particular bias, one which we may safely say that it will never cause its owner to be confined as a madman, tortured as a heretic, or crucified as a blasphemer. Also, on the other hand, it will never cause him to be applauded as a prophet, revered as a priest, or exalted as a king. And the blessings are happiness and mediocrity." Thank you very much for your attention.